Hey there, and welcome. Today, we're diving into the pretty wild world of plant cloning. Where scientists try to make perfect copies of plants, but sometimes end up with something totally unexpected. And that something might just be revolutionary.
So think about it. What does happen when a perfect copy isn't so perfect? We usually think of cloning as, hitting Control-C, Control-V, an exact replica. But you're about to see that in the plant world, it's not always that simple.
Okay, so picture this. You're in a lab surrounded by these sterile jars. Inside, scientists are doing something called tissue culture. They take a tiny piece of a plant, maybe from a leaf or a stem, and they convince it to grow into a whole new plant. The goal here is pretty straightforward: make thousands of genetically identical copies of one superstar plant. But here's the twist. Even though they all start from the exact same genetic blueprint, the new plants that pop up, well, they can be different. And figuring out why that happens is what this is all about.
This little mystery brings us to what we're calling the cloning conundrum. I mean, why would a process designed to be perfect sometimes go off script and create something new?
Right, so there's an official name for this: Somaclonal Variation. A couple of scientists, Larkin and Scowcroft, came up with the term back in 1981. It basically describes all the genetic and even physical, what we call phenotypic, differences that just show up in these clones that are supposed to be identical twins. So if the original blueprint is perfect, what's causing all these changes? Well, you might be surprised to hear that the answer isn't some outside contamination, it's the lab itself.
Yeah, it turns out the whole cloning process is actually pretty stressful for plant cells. As you can see here, everything from where they take the plant tissue to the stuff they grow it in puts the cells under a ton of pressure. And all that pressure leads to what's called oxidative stress, which is right at the heart of this whole thing. Let's break that down a little. First, where the tissue comes from, the explant, really matters. A cell from a mature leaf is more likely to change than one from a new shoot. Also, the longer you keep the cells in that lab culture, the more chances there are for mutations to pop up. And get this, even the hormones they use to make the plants grow, like one called 2,4-D, can sometimes mess with the DNA. All of this stress creates these nasty little molecules called free radicals that can directly damage the plant's genes.
And that damage really comes in two main flavors. On one hand, you have genetic changes. These are permanent, heritable changes to the DNA code itself. It's like going into a book and literally rewriting a word. On the other hand, you have epigenetic changes. Now, these don't change the words in the book, but they change how the book is read, kind of like using a highlighter or putting a sticky note on a page to turn certain genes on or off. And these changes can sometimes be reversed.
Okay, so we've got all these tiny changes happening at a microscopic level. How on earth do scientists actually find them?
Well, they have a whole toolkit for this. Sometimes it's easy. The changes are morphological, meaning you can literally just see that a plant looks different. Other times, they have to dig deeper, analyzing its internal chemistry or even putting its chromosomes under a microscope. But the real game changers are the molecular tools. And this is what that looks like. You can think of this as a sort of DNA fingerprint or barcode. Each of those vertical lanes is a different plant. See how some of the black bands are in slightly different places? Bingo. That's the Somaclonal Variation. It's an amazing way to spot these hidden genetic differences, even in plants that look completely identical from the outside.
So this brings us to the most important question, really. Is all this variation a mistake in the system, a bug that needs to be squashed? Or is it actually a hidden feature?
For a lot of folks, it's definitely a bug. I mean, if you're running a business trying to sell thousands of identical banana plants, any unexpected changes are a total disaster. It completely undermines the whole point of cloning, which is consistency. But, and this is a huge but, for plant breeders, these random errors are an absolute goldmine. They basically create new genetic diversity out of thin air. It's like a shortcut to developing new traits that might otherwise take decades of slow, traditional breeding to achieve.
And this isn't just some theory. Check out these real-world examples. A sugarcane clone that suddenly became resistant to major diseases, a new sweet potato that had a richer, more stable skin color, new types of wheat and maize with way higher yields. Every single one of these came from these so-called mistakes. And it gets better. Scientists aren't just waiting around for these happy accidents to happen. They can actually steer the process. As this shows, they can deliberately expose the cells in a petri dish to some kind of stressor, like a toxin, and then they just select the ones that survive. These survivors are the ones that have randomly developed a useful new trait, like resistance. It's a powerful way to fast-track innovation.
This quote from the research really just nails it. It says that somaclonal variation is a valuable source of genetic variation. In other words, what started out as a frustrating problem for one group of people became an incredible opportunity for another.
So what's the big takeaway here? When you step back and look at the whole picture, it's a really fascinating story.
It really boils down to this. We tried to make perfect copies in a lab, but the stress of that very process created unexpected changes. And while those changes are a headache if you want uniformity, they turned out to be an incredible gift for making our crops better. The bug became a feature, a really powerful one that's helping us create stronger, better food for the future.
It really leaves you with a pretty big thought, doesn't it? In our search for perfect control, we accidentally stumbled on the power of random imperfection. So maybe, just maybe, harnessing nature's random errors isn't just a clever trick. Maybe it's one of our most important tools for building the future of food.